After studying this module, you will be able to know what is doping, learn the rules related to doping, and identify the drugs which are abused in sports. Abuse of drugs in field of sports is often called as doping. In other words, the use of banned performance-enhancing drugs in sports is generally denoted to as doping, particularly by the organizations regulating sporting competitions. The use of drugs to augment the performance of the athlete is considered immoral by most of the international sports organizations, including the International Olympic Committee or IOC. The International Olympic Committee or IOC is a Swiss non-profit, non-governmental organization centered in Lausanne, Switzerland. Pierre Baron de Coubertin created this committee on 23rd June 1894 with Demetrius Vikalis and was its first president. The International Olympic Committee is the highest authority of the modern Olympic movement. The reasons underlying the prohibition of these drugs are mainly the health risk of these performance-enhancing drugs, the fairness of opportunity for athletes, and a standard effect of drug-free game for the public. Anti-doping authorities opine that using performance-boosting drugs goes up in contradiction of the spirit of sport. The abuse of drug is an attempt to enhance the performance in human sporting competitions and as well as in animals, for example, horse racing, etc. The toxic effects of drug are less difficult to ascertain, but the conclusions drawn from the available data are often circumstantial. Nevertheless, there is enough indication of the harmful effects when certain drugs are misused to justify their prohibition from sports competition. The chief controlling body in human sports is the International Olympic Committee or IOC. However, since 1999, doping issues have been taken over by the World Anti-Doping Agency or WADA. In WADA is an autonomous foundation shaped through the collective efforts led by the International Olympic Committee. WADA has set up on November 10, 1999 in Lausanne, Switzerland. According to WADA, the legal description of doping is the violation under the World Anti-Doping Code covers aspects such as the presence of a prohibited substance or metabolites or marker in an athlete's specimen, refusing to provide a sample or evading sample collection, tampering or attempting to tamper with any aspect of doping control, possession or trafficking of prohibited substance. WADA annually issues a list of barred drugs as an international standard. This list covers substance and methods prohibited from use by the athletes. Next is list of prohibited drugs as per WADA. In this table of the list of drugs which are not allowed in sports as per the WADA regulations, on one column we have the category of drugs and on another column we have shown the substances. For stimulants it is adrenaline, amphetamine, methemphetamine, penetrazole, phenmetrazine, phendimetrazine, etc. For narcotics, it is buprenorphine, dextroamide, diamorphine or heroin, fentanyl and its derivatives, hydroxymorphone, methadone, morphine, oxycodone, oxymorphone, pentazocine, and pethidine. In case of endogenous and anabolic agents, it is androstenediol, androstenedione, dihydrotestosterone, prasterone, testosterone, and metabolites and their isomers. In case of other anabolic agents, the substances are clenbuterol, selective androgen receptor modulators or SARMs, Typology, zeranol, and zilpatrolol. For masking agents, the substances are diuretics, epitestosterone, probenecid, alpha reductase inhibitors, plasma expanders, and materials with like biological effect. Next are the beta blockers, which includes acebutolol, alpranolol, atenolol, betaxolol, bisoprolol, bunolol, carteliol, carvedilol, ciliopropol, esmolol, labetalol, levobunolol, metipranolol, metoprolol, nadolol, oxpranolol, pindolol, propanol, soltalol, timolol and their related compounds. And the prohibited methods are the enhancement of oxygen transfer, chemical and physical manipulation including the intravenous infusion and sample tampering, and lastly the gene doping. For most substances, mere existence of the substance or a diagnostic metabolite in the biological fluid sampled constitutes an offense. But for some substances, there is a reporting threshold. If drug is detected but it is below the reporting threshold, no offense is deemed to have occurred. The WADA list also recognizes that some anabolic agents and hormones are produced endogenously. The underlying rationale is that an offense is considered to have occurred if one of the prohibited drugs has been used in competition sports such as shooting, billiards, gymnastics, etc. Alcohol is prohibited in competition for sports such as motorcycling, powerboating, archery, etc. Next is the typology of drugs misused in human sports. Here are some of the categories of drugs that I will be telling you about, which are generally misused in games. First is the anabolic androgenic steroids, which include 1-androstenediol, 1-androstenedione, bolandiol, calistrol, clostibolol, danazol, furazabol, mestanol, and mestosterone, etc. Next class is the peptide hormones such as the chorionic gonadotropin or CG and luteinizing hormone, also known as LH, and their releasing factors, corticotrophins, growth hormones, etc. Third class is the diuretics and other masking agents, which includes the masking agent comprising of the plasma expanders, example glycerol, intravenous administration of albumin, dextran, etc. Diuretics includes acetazolamide, canceron, clotadione, furosemide, indapamide, metolazone, etc. The metabolic modulators including insulin, peroxisome proliferator activated receptor agonist, etc. Next is the prohibited methods. There are certain methods which are banned in sports, and few of them we will discuss in this module. First is the manipulation of blood and blood components. The procedures that are prohibited in sports under this category are the administration or reintroduction of any quantity of autologous, homologous or heterologous blood or red blood cell products of any origin into the circulatory system, artificially enhancing the uptake, transport or delivery of oxygen, including but not limited to efaproxyrol, that is RSR13, perfluorochemicals, and the modified hemoglobin products, example hemoglobin based blood substitutes, microencapsulated hemoglobin products, and excluding the supplemental oxygen. Any form of intravascular manipulation of the blood or blood components by physical or the chemical means. Next is chemical and the physical manipulation, which includes the following procedures that are prohibited. Under this category, they are tampering or attempting to tamper in order to alter the integrity and validity of samples collected during doping control. These include but are not limited to urine substitution and or the adulteration, example the proteases. Intravenous infusions or the injections of more than 50 milliliter per 6-hour period, except for those legitimately received in the course of hospital admissions or the clinical investigations. Third topic is the gene doping. The potential to enhance the sports performance is the gene doping, and various procedures are prohibited. The transfer of polymers of nucleic acid or nucleic acid analogues, the use of normal or genetically modified cells. Next, students, we will study about the substances prohibited in particular sports. There are few substances use of which is not allowed in some particular sports, and some of them are, starting with the first, we have the alcohol. The usage of alcohol, especially ethanol, is prohibited in competitions in the succeeding sports only, such as karate, air sports, archery, automobile, motorcycling and powerboating. Breath analyzer method or blood test can assist recognize their concentration in the blood. The doping violation threshold for the concentration of alcohol in the blood is 0.10 grams per liter. Next are the beta blockers. Beta blockers are prohibited in competition only in the sports such as archery, automobile, billiards, darts, golf, shooting, snowboarding. Beta blockers includes but are not limited to acebutolol, cartiolol, atenolol, esmolol, betaxolol, timolol, nadolol, etc. Next is the sampling. Sample collecting procedure must be in accordance to both the scientific and legal aspect, which has been discussed in this module. First is the health of the individual being sampled must be safeguarded. Incorrect labeling, contamination of the collected sample or sample switching must be avoided. The rights of interested parties, generally the owner and trainer of an animal or the individual or team in human sport, must be safeguarded against any error by the analyst. First, we will study about the human sampling. The World Anti-Doping Code International Standard for Testing describes the procedure or the process for sample collection. Two matrices specified for testing are urine and blood. Urine is the primary matrix used for testing because it is considerably less invasive in terms of sample compared to blood, offers relatively low health risk and safety compared to blood in terms of sampling and analysis. Blood may be sampled and used instead of urine. Blood is used as a test matrix when detecting blood transfusion. The principal advantage of a blood sample is that its integrity is easier to safeguard because usually it is collected by a doctor or veterinary surgeon experienced in the particular procedure. Drug concentration in blood are interpreted more easily than those in urine, and certain drugs that are not excreted in urine in detectable quantities, example include reserpine or human growth hormone, can be detected in blood easily. Next is the Sample Collection Authority. The organization that is responsible for the collection of samples in compliance with the requirements of the International Standard for Testing and Investigations, where the testing authority itself or another organization, for example a third-party contractor, to whom the testing authority has delegated or subcontracted such responsibilities. Third is the sample collection equipment. The sample collection equipment shall as a minimum consist of that is for urine sample collection vessels for collecting the sample as it leaves the athlete's body, suitable kit for storing partial samples securely until the athlete is able to provide more urine, and sealable and tamper evident bottles and lids for storing and transporting the complete sample securely. For blood sample collection, needles for collecting the sample, blood tubes with sealable and tamper evident devices for storing, for transporting the sample securely. Next, we will study about the detection of drugs. There are various sophisticated techniques which can be deployed for the detection of drugs in blood or urine. Two of the frequently used techniques are the solvent extraction procedure and TLC, or the thin layer chromatography. Drugs are extracted with dichloromethane and 2-propanol mixed in the ratio of 3:1, and aliquots of the extract are applied to the TLC plate. The developed plates are examined under UV light and are then subjected to a range of visualizing reagents. Steroids are extracted from urine using ethyl acetate after adding saturated sodium borate solution and washing the extract in 15% sodium sulfate in sodium hydroxide solution. Then the extract is allowed to run on the TLC plate. First here we will discuss about the solvent system. The solvent system comprises of chloroform is to ethyl acetate is to methanol in the ratio of 80:45:5 respectively. The plate is dried and then rerun in Davidon reagent. For visualizing reagents, sulfuric acid is to ethanol in the ratio of 50:50 is used as the visualizing agent. It also requires heating with hot air blower to develop the dark spot. Next is the techniques used for the confirmation test. There are few techniques which are employed for performing the confirmatory test, which include GCMS, that is gas chromatography and mass spectroscopy, LCMS, and third is the immunoassay or the HCG, that is the human chorionic gonadotropin hormone. Then about the threshold analysis. Threshold may be set either in the rules for laboratory reporting or as a penalty threshold. Each laboratory needs a protocol to determine whatever the specimen threshold has been exceeded or not. Now students, let us summarize this module. Drug abuse in sports is generally referred to as doping. The abuse of drug is an attempt to enhance performance in human sporting competitions and as well as in animals, for example, such as horse racing, etc. According to WADA, the legal definition of doping is a violation under the code cover aspects such as the presence of a prohibited substance or metabolites or marker in an athlete's specimen, refusing to provide a sample or evading sample collection, tampering or attempting to tamper with any aspect of doping control, possession or trafficking of prohibited substance. For some substance, there is a reporting threshold. If drug is detected, but it is found to be less than the reporting threshold, no offense is deemed to have occurred. Sample collecting procedure must take into attention both the scientific and the legal aspects as well. In human, urine and blood are the most recommended samples for collection. For examination of presence of drug in samples, several techniques are used or can be employed. Example, TLC, LCMS, GCMS, immunoassay, etc.